Right. In this lecture, we are going to look at excitation-contraction coupling, which are some of the first steps of how a muscle contracts. So let me share my screen. All right, so what we're really asking here, and what we're going to look at over this lecture and the next, is how does the action potential of a motor neuron result in the contraction of a muscle? And what we're going to see is that there are basically four steps, four big stages to that. The first one we can refer to as excitation. This is where you have communication between the neuron and the muscle cell. And what this leads to is the excitation of the muscle cell. And remember, what excitation means is actually that an action potential is going to travel across the surface of the muscle cell. So we've been learning about action potentials in neurons, but muscle cells are also capable of propagating an action potential across their plasma membrane. Okay, so that's the excitation phase. Next, we have what's called excitation-contraction coupling. And this is where the action potential of the muscle cell is going to be converted into actually activating the proteins inside the sarcomere, which is then going to lead to contraction, where you have movement of those proteins that is going to cause the muscle to develop tension, and the muscle may then shorten. And contraction works via what's called the sliding filament theory, which is basically a description about how the thick and thin filaments within the sarcomere are going to interact with each other. And then our last stage is relaxation, where the muscle cell is going to return to its resting length. So in this video, we're going to focus on first excitation, and then excitation-contraction coupling. And then the third and fourth stages will be in a subsequent video. So the first thing that we need to look at with excitation is we need to look at this area called the neuromuscular junction. We can shorten that to NMJ. And the NMJ is essentially a synapse between a motor neuron and a muscle cell. So we've been talking about synapses previously, looking at them as connections between two neurons. Now we're looking at the connection between a motor neuron and a muscle cell. All right, so here is our motor neuron, right? Here is its axon, and here we're looking at the axon terminal, or what can also be called in this image the synaptic knob. Okay, so this area right here. What we're looking at here, this is the muscle cell. And specifically, this is a region of the muscle cell called the motor end plate. And so it's the part of the sarcolemma, right, the part of the plasma membrane of the muscle cell that is associated with the neuromuscular junction. Okay, so that is right next to the axon terminal of our motor nerve. And when we look at, if we look at the sarcolemma right here at the motor end plate, what we're going to see is that it is covered in these little chemically gated ion channels. These are shown here, these little blue guys right here. So these are chemically gated ion channels. And specifically, these are ion channels that respond to the chemical acetylcholine. Okay, and acetylcholine is a neurotransmitter that is found within synaptic vesicles inside the motor neuron. Okay, so what's going to happen, if we look at this image right here, right, an action potential is going to come down the axon of that motor neuron, and it's going to reach the end of the axon. And similar to synapses that we've looked at before, when that action potential reaches the end of the axon, it's going to cause voltage-gated calcium channels to open within that axon terminal. When that happens, calcium is going to enter into the neuron. And when calcium enters, it's going to cause those synaptic vesicles, these guys right here, they're going to release acetylcholine into the synapse. So that's what we can see in this image. We have these synaptic vesicles, and we can see that they are opening and releasing acetylcholine in green here into the synapse. Acetylcholine is then going to bind to these acetylcholine receptors, which again are found within the sarcolemma of the muscle cell. All right, so here now we've zoomed in and we can just see one of those acetylcholine receptors, again, sitting within the sarcolemma, within the plasma membrane of the muscle cell. So acetylcholine is going to bind to that receptor. And remember, this receptor is a chemically gated ion channel, which means that when this chemical, when acetylcholine binds to it, it is going to change shape, as we can see in this image here, and it's going to open up. And this is actually a rather non-specific channel, meaning that it allows for the diffusion of both sodium and potassium cations. So based on what we've talked about before about these concentrations of these ions, we would expect, and what we will see is that sodium is going to rush into the cell and some potassium is going to exit the cell. But what happens is that more sodium enters than potassium leaves. And so you have this inward rush of positively charged sodium ions. So you're going to wind up depolarizing the muscle cell. And we call this depolarization, we refer to it as an end plate potential. Okay, think of it as kind of similar to a graded potential that we would see in a neuron, but it's happening at the end plate of a muscle cell. So we call it an end plate potential. All right, now that depolarization, similar to a depolarization that we would see with a graded potential, is going to trigger an action potential in the muscle cell. And again, this will happen when it reaches threshold. So there will be a threshold for that cell. And when the membrane potential reaches threshold, you're going to trigger an action potential. And the action potential of a muscle cell is going to work the same way that it does in a neuron. So you're going to have voltage-gated sodium and potassium channels that are going to open sequentially across the surface of the muscle cell membrane. Causing a rapid depolarization and repolarization. Okay, so the same way that it does traveling down the axon of a neuron, the action potential is going to travel across the surface of our muscle cell. And as it does, we can see in this image here, these little arrows are indicating the movement of that action potential. So it's going to be propagated across the sarcolemma. And remember that the sarcolemma extends down into these tubes called T-tubules that actually penetrate down into the middle of the cell. So the sarcolemma that extends down into those T-tubules, the action potential is going to continue to be propagated down across that sarcolemma. So the action potential is going to travel down into these T-tubules, so it can actually penetrate into the middle of the cell. And that's really important. Remember that the T-tubules, so we can see the T-tubule here, and then we can see right here the terminal cisternae of the sarcoplasmic reticulum, which remember is full of calcium ions, these little sort of beige circles here. So we're going to see in this next step, what happens is, as the action potential propagates down the T-tubule, it is going to open voltage-gated calcium channels that are found in that T-tubule. Now what's interesting is that those voltage-gated calcium channels are actually connected, physically connected, to mechanically gated calcium channels in the sarcoplasmic reticulum here. And so what's going to happen is, as the action potential travels down that T-tubule, it's ultimately going to trigger these channels to open in the sarcoplasmic reticulum. And so the end result is that calcium, these calcium ions that are stored in the sarcoplasmic reticulum, are going to be released into the muscle cell. Okay, so the end result, right, you have an action potential that travels down your motor neuron, and that's going to trigger acetylcholine to be released at the neuromuscular junction. That is going to trigger an end plate potential, which if it reaches threshold will then trigger an action potential. And that action potential, as it spreads across the muscle cell, is going to trigger all of this calcium to be released within the muscle cell. So as I said, right, the sarcoplasmic reticulum is going to release calcium ions into the sarcoplasm. And this is very important because what's going to happen is that once you have calcium ions within the sarcoplasm, those calcium ions are going to bind to troponin. And remember that troponin is found within thin filaments. So it binds to troponin. Troponin itself is attached to tropomyosin. And when calcium binds, it's going to cause troponin to change shape, which is going to cause the tropomyosin to be moved off of the myosin binding sites on the actin molecules. So now these binding sites are now open, which means that myosin is going to be able to bind to actin. Okay, so that's sort of the ultimate result of releasing calcium into the cell. And in our next lecture video, we will look at what then is going to happen between the actin and myosin to cause contraction of muscle.